Hi, it's Moser, and I am going to walk through the learngeneticsutah.edu gel electrophoresis simulation because it does not work on mobile devices such as iPads. You're holding a small plastic tube with some clear liquid in it, and you've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? Press forward to continue. DNA strands were as big as your shoelaces. You could sort them by hand into groups and measure them. But DNA strands are molecules so tiny you can't see them even under most microscopes. Is there a way to sort and measure DNA strands in your tube even though you can't see or touch them? There is. It's called gel electrophoresis. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like protein. How does it work? The gel is the filter that sorts the DNA strands. It's like a sponge made of Jell-O with many small holes in it. We place DNA samples into the holes at one end of the gel. Electrophoresis is how we push the DNA strands through the gel filter. By adding an electrical current, we can make the DNA move. Short strands move through the holes in the gel more quickly than long strands. Over time, the shorter strands in the sample will move further away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. In this way, the DNA strands in the sample sort themselves. Staining the sorted groups of DNA makes them visible to the naked eye. Although we cannot see a DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. Now it's your turn to run a gel.
Interesting.
Now it's your turn to run a gel. That's scientist slang for gel electrophoresis. Follow along with the steps shown above. We'll press forward to continue. Here's what you'll need to make a gel. Powdered agarose, a buffer, a flask, a microwave, a gel mold, and a gel comb. Making an electrophoresis gel is a lot like making Jell-O, but you won't want to eat this type of gel. So we're going to click and drag the spoon over to the flask and put a small amount of agarose into the gel. Agarose is a dried powder similar to Jell-O. Add some liquid buffer to the flask. Buffer is a saltwater solution that will let electrical charges flow through the gel. There we go. Now we've loosely placed plastic wrap over the top of the flask to prevent the liquid from boiling over. Open the microwave by pressing the button. Place the flask containing the buffer and agarose mixture inside the microwave and heat until the agarose melts into the buffer. We've removed the plastic wrap from the top of the flask. Pour the melted agarose into the mold, notice that the mold has tape on each end to hold the gel in place. Place the comb into the gel on one end, and the comb will make little holes in the gel that will hold the sample. Let the gel cool and solidify. This usually takes about a half an hour, but we'll speed up the clock for you. As the gel cools, tiny holes will form in it. Carefully remove the comb, leaving empty wells for the DNA. Your gel's ready now. Now it's time to set up the electrophoresis box. You'll need the gel you just made, an electrophoresis box, and another bottle of buffer. Pour the buffer into the electrophoresis box. There we go. Place the gel, still in the mold, into the electrophoresis box. We've removed the tape from each end of the gel. You're ready to load the DNA sample into the gel. Here's what you'll need: loading buffer, the tube of DNA size standard, a micropipetter, and the electrophoresis box containing the buffer, gel, and pipette tips. With a clean pipette tip, use the micropipetter to suck up some loading buffer, then add it to the DNA sample. DNA samples are prepared in a clear liquid solution that would be hard to see if you tried to load it directly into a well. The loading buffer contains a dye that makes the sample easy to see. It's also slightly goopy. This makes the DNA sample thicker so that it will drop into the well instead of floating away. The DNA size standard already contains the loading buffer. Next, you'll use the micropipetter to transfer the DNA sample from the tube into the well of the gel. First, suck up some of the DNA sample into the pipette tip. Next, you'll use the micropipetter to place the sample in the holes. Nice job! In real life, loading the samples into the wells takes some practice, so don't be disappointed if you miss your target on the first few tries. Using a clean pipette tip, use the micropipetter to suck up the DNA standard. Transfer the DNA size standard into the microwell. It's time to turn on the electricity and run your gel. When you turn on the power, the black end will generate a negative charge. The red end will generate a positive charge. Together, they'll pass current through the gel DNA that has a negative charge. To move the DNA through the gel, you must put the black cord closest to the wells. Press forward to continue. Plug the black cord from the electrophoresis box into the matching outlet on the power supply. Turn on the power supply. Your gel is off and running. Let's look into the gel box and see what's happening. Check for tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. These bubbles are your proof that a current is running. Repelled by the negative charge, the DNA moves through the gel towards the positive charge at the other end. Short DNA strands move through the holes in the gel more quickly than long strands. Over time, the shorter DNA strands will migrate further from the starting point than the longer strands. We can't actually see the migrating DNA bands, but we can see the blue dye from the loading buffer as it migrates. You finish running your gel. We've taken the gel mold with the gel in it out of the electrophoresis box. The time has come to analyze your sample and estimate the length of the DNA strands. First, you need to stain the DNA in your gel using the DNA staining solution. The stain is a chemical called ethidium bromide, which binds to the DNA and shows up under fluorescent light. Although we can't see single DNA strands, we can see large groups of stained DNA strands. These groups will show up as bands in the gel. Drag the gel out of the mold and put it into the DNA staining solution. Because ethidium bromide binds to DNA, it can damage the DNA in your cells. If you stain a gel in real life, wear gloves and avoid direct contact with the staining solution. So we're going to drop that in there. It takes about a half an hour to stain the DNA in the gel. We'll speed up the clock for you. Remove the gel from the staining solution and place it on the ultraviolet light box. Turn on the UV box. Now you can determine the approximate lengths of the DNA strands in your sample. Compare the bands from the DNA sample with the bands of known length from the size standard. Enter the estimated length in base pairs for each band in your DNA sample. Electrophoresis cannot tell us the exact DNA length of strands, just a good estimate. So give it your best guess based on the DNA size standards. So this one looks to me like it's about 6,000 base pairs long. Whoops. And this one, I'll say, is maybe 3,200 base pairs long. And this one, maybe 1,500 base pairs long. Let's see what it thinks of my estimations. One or more of your answers was incorrect. The estimated length in base pairs of each band in your DNA sample is based on how they line up with the bands of known length in the DNA size standard. The top band is approximately 6,000 base pairs, the second is approximately 3,500 base pairs, and the third is approximately 1,500 base pairs. Congratulations! You've just run your own DNA gel electrophoresis experiment. So the big thing is, this is what a DNA gel electrophoresis produces. This is the finished product, and it can be used not only to sort individual bands by size, but we can compare DNA from two individuals using a gel electrophoresis band pattern and see how much DNA they probably have in common. That's it.